Okay, Assalamualaikum and hi everyone. So my name is Nick Saman Farisi and I'm taking Bachelor of Science with Education at University Malaya. So today we will learn about cell structure and cell organizations from Form 4 Chapter 2. So what we will learn is how to define the term organelles, how to identify the component of organelles in animal cell and plant cell. Next is how to state the function of the cellular components in an animal cell. And lastly, state the function of the cellular component in a plant cell. So about cell theory, all living things are made up of cells. Second is cells are the smallest working unit of all living things, and all cells come from pre-existing cells through cell division. So what are the cells? A cell is the smallest unit that is capable of performing life function. So this is the example of cells, which is at the left side you see animal cells, and at the right side you will see the plant cell. So the living component of cell has protoplasm, which is cytoplasm plus nucleus, surrounded by the plasma membrane, and plant cells have an outer boundary called the cell wall. Only plant cells have cell wall. So this is the similarity and difference in plant and animal cells. You can see plant cells and animal cells. At plant cell, you can see chloroplast, cell wall, and large vacuole. However, you cannot see chloroplast on animal cells and cell walls and cell wall at animal cells. So component of the cells divided to two categories, which is organelles and non-organelles. First is organelles, which is it has ribosome, smooth and rough endoplasmic reticulum, vacuole, nucleus, mitochondria, chloroplast, which only have at animal at plant cells, Golgi apparatus, lysosomes, centrioles. Next is non-organelles, which is cell walls, which is only have at plant cells. Next is plasma membrane and cytoplasm. So we will look the structure of non-organelles, which contain three organelles, or three non-organelles, which is first plasma membrane. So you can see here the structure of plasma membrane. The characteristic is thin, semi-permeable membrane. Next is this plasma membrane consists of protein, which is the purple one, these proteins. Next is phospholipid bilayer, as you can see here, and outer boundary of the cells. So the function is to regulate and control the movement of substances in and out of the cell due to its semi-permeable characteristic, and protect the cell by separating the contents of the cell from its environment. Next is cytoplasm. So the characteristic is a jelly-like medium containing water and mineral salt, as you can see here, these pictures. And it has contains organelles. So the function is acts as a medium where biochemical reaction and most living processes occur within the cell, and provide the organelles with substances obtained from external environment. And the last one is cell wall. The characteristic is rigid outer layer of plant cell which is made of cellular fibers and fully permeable. It gives a rigidity to the cells. Together with the central vacuole, it provides support to the cell. It supports the cell to maintain shape of the plant cells and protect plant cells from rupturing due to the movement of excess water into the cell. So next is about organelles. We have 9 organelles here, which is vacuoles, nucleus, mitochondria, ribosome, endoplasmic reticulum, Golgi Apparatus, lysosome, centriole, and chloroplast. If you want to remember these organelles, you can give an acronym such as V and M until C, so you can recall it back when you are in examination. So first, what is organelles? Organelles is a subunit in the cytoplasm that carry out a particular function. So each of these organelles have their function, which is for the first is nucleus. You can see here the image of nucleus. The characteristic is spherical with double membrane. It consists of nuclear envelope, which is this, the bigger one, nuclear plasm, which is this, nucleolus, the middle, and the chromatin. And this is the nuclear pore. As you can see here, we have pore around the nuclear envelope. So the nucleus DNA, which is consistent of genetic material, is carried in the chromatin in the nucleus, in the nucleolus. So the function is to control all activities of the cell, and nucleolus is involved in the synthesis of ribosome and ribonucleic acid, which is RNA. The outer membrane of the nuclear envelope is continuous with the rough endoplasmic reticulum, RER. Here you can see the nuclear envelope continues with the rough endoplasmic reticulum. All the cell has nucleus, but mature red blood cell and phloem sieve elements does not have nucleus. Next, we will talk about the plasmic reticulum, which consists of extensive flattened membrane folded repeatedly to form a large network of interconnected tubes and sacs. Each membrane is the continuation of the nuclear membrane, and endoplasmic reticulum have two types, which is rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum here is attached with a ribosome, whereas the smooth endoplasmic reticulum does not have the ribosome attached to the structure. So first, about the rough endoplasmic reticulum, the characteristic is made up of flattened membrane disc. Next is rough ER, which has a ribosome on its surface. As you can see the dots here, it is ribosome attached to the structure. And the outer membrane of the nuclear envelope is continuous with RER. So the function is to transport protein, which are made by the ribosomes on its surface. Next is smooth endoplasmic reticulum. The characteristic is made up of tubular cavities. Smooth ER does not have ribosome on its surface and occurs away from the nucleus. The function is to synthesize lipids, center for detoxification of drug, and make steroid. This is the type of a lipid steroid. So for the next organelle is Golgi Apparatus. The characteristic is consists of a stack of membrane-bound sacs with budding vesicle around it. As you can see here, stack membrane brown, elongated. So the function is act as a center for processing, packaging, and transporting macromolecules such as protein and carbohydrate. So remember, the Golgi processing, packaging, and transporting macromolecules such as protein and carbohydrate. Next is mitochondria. This is the structure of mitochondria. As you can see, you can look at this in a membrane and outer membrane. Mitochondria have inner membrane and outer membrane, and this is cristae. And inside the cristae, we have matrix, and this is the intermembrane space. So the characteristic is rod shaped with a double membrane. As you can see here, inner and outer. Outer membrane is smooth and regular, while inner membrane is folded to form cristae. The function is known as the powerhouse of the cell, which is release energy as it is the site for cellular respiration, and energy generated or released in form of ATP. Next is lysosomes. The characteristic of lysosome is membrane-bound vesicle found in animal cells only. So you cannot find lysosome in plant cell. They only appear in animal cells only. So the function is first contain enzyme which control breakdown of protein and lipid. Next is contain enzyme that digest aged or defective cells components. It also self-digestion of a cell, and in certain unicellular organisms, lysosome fuse with food vacuole and dispense their enzyme into these vacuoles to digest the content of the vacuole. So you can see here the lysosome, which has membrane, and inside it consists of protein or enzyme. Next is centriole. So the characteristic is consists of two cylindrical body structure arranged at right angle to one another. And these organelles only found in animal cells, which is you also cannot find this organelle on plant cell. So the function is for the formation of spindle fiber during cell division. Next is chloroplast. The characteristic is dish-shaped organelle with double membrane, which also has inner and outer membrane. That's why we call it as double membrane. So it consists of grana within stroma, which is here. Within the stroma, we have the grana. Each granum contains chlorophyll pigment. Each of these granum consists of chlorophyll pigment, and the green pigment of chlorophyll give plants their color. So the plant's color is green, and the green pigment of chlorophyll that give the color. So the function is carry out photosynthesis, and chlorophyll capture the energy of sunlight and convert light energy into chemical energy during photosynthesis. Next is vacuole. For this organelle, you only can find it on plant cells. Same as these organelles, which is vacuole. It has cavities filled with cell sap surrounded by a semi-permeable membrane called tonoplast. So the function is contains water, sugar, and dissolved materials. It is to maintain rigidity of cell in plant. You can see here the big one. This is the central vacuole in the plant cells, and not animal cells have small vacuoles that are temporary. So it's only temporary. Example, amoeba and bacteria. So looking back to this picture. So if you are on examination, you can use this structure. You can draw the structure. It is acceptable, and we look back to the organelle in plant cell and animal cells, which is this is mitochondria in plant cells. The Golgi apparatus is the orange one, the plasma membrane, and the cytoplasm. The cell walls only appear in plant cells. We have plant cells and not in animal cells. The centriole also only have animal cells and not in plant cells. So if you are going to give an answer about similarities and difference, you can use this as your reference. So I think that's all from me. I hope you learn, you achieve the learning outcome, which is you are able to define the term organelles, able to identify the component of organelles in an animal cells and plant cells, and able to state the function of the cellular components in an animal, state the function of the cellular components in a plant cells. So that's all from me. Thank you.